Dear students, welcome to Tumala online classes. In the last video, we were starting with the mitotic cell division, right? So, mitosis, it is one type of cell division in which there we made two phases. One is interphase and second one is M phase. And in interphase, so it is also known as a preparatory stage, right? And in M phase, there we come across four stages, and they are prophase, metaphase, anaphase, as well as telophase. These are the phases that we come across there. So after these stages, there we go to the cytokinesis, right? So the blueprint of this one, let us put first, and afterwards we'll start to study one by one the stages here. See here, mitosis is one type of cell division which is divided into two phases. One is called as interphase and second one is called as M phase, right? Interphase as well as M phase that we come across over here. So interphase we have already studied there. In M phase, what we come across? In M phase, so here nuclear division, we study nuclear division. It is also called as karyokinesis, and so second one is cytokinesis, which is also known as cytoplasmic division, right? So first of all, we will have to go through this nuclear division. First, see here, nuclear division. Nuclear division, in which there we come across four stages. Four stages. What are those? One is prophase, second one is metaphase, and third one is anaphase, and the fourth one is telophase. So these are the four stages that we come across over here. And after this nuclear division, we will go to the cytokinesis, that is cytoplasmic division. Okay, so these are the two stages that we come across here. First of all, we will start with the nuclear division. And one more thing we must be aware of, already I have mentioned. So this mitotic cell division is usually taking place. So it is usually taking place in somatic cells. Somatic cells. Cells may be haploid in nature, diploid in nature, or polyploidy in nature. So whatever the ploidy of the cell, so it doesn't matter, but so the cell division occurs over here mitotically, right? In somatic cells. In somatic cells, it must be very clear to you people. Okay. So this is a blueprint of the mitosis. So let us start with the first stage of nuclear division. What is that? Prophase. What happens in prophase? Let us see. Now, prophase. In prophase, what happens? Let us see. In case of animal cell as well as in case of plant cell, we will have to see here. So this is one animal cell which contains the plasma membrane, and this is nuclear membrane. Nuclear membrane, and inside which there we come across what? The chromatic network is present over here. So, and nucleolus is also present over here. In this, what happens? Let us see one by one. In this cell, initially, the condensation of this chromatic network starts. This chromatic network start to condense itself, and this prophase is longest phase in mitosis, right? It is longest phase in mitosis, so or especially in karyokinesis. So means it takes place long period. It takes place in long period. This is the fact what you should know. And here the condensation of chromatin network starts. When the condensation of chromatin network starts, that moment what happens? Very condensed thread-like chromosomes start to form here. Here the formation of a short, so thin thread-like, so chromosomes are formed over here. Here the arms of the chromosome start to coil itself, recoil itself, right? So as to form the condensed thin thread-like structures, that is chromatids. So they are called as the arms of the chromosomes. They are called as. So very thick and short, thin thread-like chromosomes start to appear in this stage. Simultaneously what happens here? Coiling of the chromatin network also starts over here, and it is called as spiralization. So as a result of spiralization, as a result of condensation, so the short structured, so chromatids start to form over here. Simultaneously, the centrioles what are present over here. So a pair of centrioles are lying right angles to each other, and these centrioles start to move apart, means both of themselves they start to move apart. Here one centriole that comes over here, and one centriole that remains over here, and so these centrioles when they start to move, so towards both the poles, this is one pole and this is another pole, simultaneously they start to produce minute microtubules. Minute microtubules start to arise from these centrioles, and they are looking to be as stars. Hence these are forming the asters. So what they form? Asters. So they start to radiate small microtubules from themselves, and both of themselves start to move apart, so towards both the poles. One remains here, another one start to move over here. So here what happens? So as a result of the radiation of these microtubules from these centrioles, so they look to be as stars, hence they are called as esters. Means here centrioles start to move towards the opposite poles. They start to move towards the opposite poles. Okay, they start to move towards the opposite pole, and simultaneously, what happens over here? So they start to form the spindle fibers, right? Initially, the spindle fibers are started to form over there. And this nucleolus gradually start to become smaller enough, right? It start to become nucleolus, start to become smaller enough, and gradually what happens? Nuclear membrane also start to disintegrate. Gradually. It start to disintegrate. See here, it start to disintegrate. Right, like this. It start to disintegrate like this. Okay. Disintegration of nuclear membrane is taking place. Gradually the formation or the condensation of this chromatid into chromosomes. So chromatin into chromosomes, that is taking place. Gradual disappearance of the nucleolus is taking place. Movement of the esters towards opposite poles is taking place. All these are the activities that take place over here initially. And at last, post-prophase it is said as. This is so prophase, initially what takes place? Post-prophase, in post-prophase what happens? Disappearance of nuclear membrane and nucleolus takes place. This leads to leaving the chromosomes directly into cytoplasm. See here, now the chromosomes, they came into the cytoplasm. Why? Because the gradual disappearance, complete disappearance almost. See here, disappearance of both of these. Now where the chromosomes are? They are in cytoplasm. Right? So this is the fact what happens in case of the prophase. Okay? So in prophase, this is the activity that is done by the nucleus itself. Simultaneously there the cell organelle is like endoplasmic reticulum, Golgi complex, they become inconspicuous, right? Simultaneously the Golgi complex and endoplasmic reticulum become inconspicuous. So very negligible. So inconspicuous they become, and simultaneously, so the esters what they form the spindle fibers, it is taking place only in case of the animal cell, not in case of plant cell. Why? Because esters are absent in plant cells, centrioles are absent in plant cells, hence the spindle formed is ancestral in case of plant cell and it is amphiestral in case of animal cell. Right? Point to be noted. Amphiestral in animal cell and ancestral in plant cell. Alright? The spindle fiber is amphiestral in animal cell. Why? Because two esters are formed over here, from which the esters are formed over here, from which the spindle fiber is formed. But ancestral in the sense that there the spindle fiber is formed without centrioles. Next stage is metaphase. In metaphase what happens? It is a short period stage, 2 to 10 minutes it takes, right? It takes 2 to 10 minutes over here. In metaphase what happens? Let us see here. This is the cell. So where the nuclear membrane is totally absent there. So the esters they have become to the poles respectively, and these have formed already the spindle fibers over here. Okay. So here what happens over here is if the chromosomes are there, they are start to coming towards the equatorial line. This is the line called as equatorial line. So all the chromosomes are brought to the equatorial line with the help of these spindle fibers. With the help of these spindle fibers. How they are brought to the equatorial line is the mat over here. Okay. So they are lying just like this. This is the centromere of our chromosome. So these are the chromatids of the chromosome here. So they are attached. They are lying on the equatorial line like this. A single diagram I have drawn for your convenience purpose. Okay. What happens here? So across this cell, all the chromosomes are lying initially. So in the post-prophase stage, in the pre-metaphase stage, what happens here is, so the attachment of these chromosomes to the spindle fibers take place. Before that one, the formation of this spindle apparatus is formed. Here, formation of spindle apparatus has completed. After this one, after its completion, what happens here? Spindle apparatus is formed over here, and chromosomes attached to spindle fibers, right? How do they attach to the spindle fibers? So whenever you come across one chromosome, you know this is one chromosome to which a centromere is present. So chromatids, these are chromatids. Chromatids are attached to the centromere, right? It is also called as primary construction, to which C-shaped, disc-shaped structure is so attached on the centromere, and it is called as kinetochore. We know it. So from these one, there you come across this kind of fibers or the microtubules. These fibers are called as chromosomal fibers. What these are? Chromosomal fibers. These chromosomal fibers are attached to the spindle fibers. This is how the chromosomes attach to spindle fibers with the help of chromosomal fibers arise from kinetochore, right? So this is how the chromosomes are hanged to the spindle fibers formed there. Okay, now we came to know how the attachment of these chromosomes that take place to the spindle fiber, and simultaneously these chromosomes are brought towards the equatorial line, right? See here, chromosomes are brought to the equatorial line and are lying on equatorial line in such a way that the centromeres of all chromosomes lie right angles to the spindle fibers. See here, the chromosomes are lying just like this, right? And one more chromosome I shall draw over here. See, this is also the centromere of one chromosome. It is lying like this. Okay, this is one chromosome. Now we came to know these chromosomes are hanged to the spindle fibers and they attach to the spindle fibers and they are brought towards the equatorial line. So what it looks to be as is, this is another centromere, this is another centromere, this is another centromere, this is another centromere. See here, all these centromeres of the chromosomes are attached to the spindle fibers here like this. These are all spindle fibers. So to which the attachment of chromosomes that take place over here. Like this, all are lying in the equatorial line. So that what happens? All chromosomes lying on equatorial line to form a plate-like structure called as equatorial plate, right? Equatorial plate is called as. So that kind of structure it forms over here. In this case, metaphase chromosomes are lying on the equatorial line formation of equatorial plate that take place over here. So this is one. And there the fibers, already I told you there are three types of fibers that we come across over here. What kind of fibers these are? One is continuous spindle fibers, one is continuous spindle fibers, and second one is discontinuous spindle fibers, and third one is chromosomal fibers. Chromosomal fibers, continuous fibers, discontinuous fibers, as well as chromosomal fibers are the three kinds of fibers that we come across here. See here, so these are the fibers that we come across here is, this is one cell, this is one ester, this is another ester. See, they are continuing from one pole to another pole, right? If they continue from one pole to another pole, then they are called as continuous fibers. They are called as continuous fibers. Sometimes what happens, some of the fibers which are making the discontinuous one, they come on here and stop themselves. They do not reach another pole, right? They do not reach another pole. They are called as discontinuous fibers, right? One thing. And chromosomal fibers, you know it better. From the kinetochore, whatever the fibers that arise, and they hang to the, or they reach to the one pole of the cell, then it is said to be as, chromosomal fibers. That kind of three fibers that we come across over here. And last point and very important point. The process of bringing all the chromosomes on the equatorial line, forming an equatorial plate, is called as congregation. Congregation. Right? What do you mean by congregation? It is nothing but bringing of all the chromosomes on the equatorial plate. So it is done by the spindle fiber. So this is called as what? So the metaphase. So these chromosomes are highly thickened one, highly thicked, darkly stained, right? These are the chromosomes which are suitable for their study itself, right? So this is one more fact to be remembered regarding the metaphase. And the next stage is anaphase. In case of anaphase, what happens? Let us see. Next stage is anaphase. It is a rapid stage of cell division in which movement of chromosomes take place. Movement of chromosomes take place towards opposite poles of the cell. See a rapid movement of the chromosomes that take place on the opposite poles that occurs in case of anaphase over here. See what happens here is already the chromosomes are hanged to the spindle fibers. This is one ester, this is another ester. One spindle fiber, this is another spindle fiber, to which the chromosomes are hanged over here, right? Now all are lying in the equatorial line. I am going to show only two chromosomes over here. This is one chromosome with two arms. This is another chromosome with two arms. See, whenever these are lying in the equatorial line, in case of anaphase, where the breakdown of this centromere that takes place, this is the centromere, the breakdown of this centromere that takes place. See here, this is one chromatid, this is another chromatid, this is chromatids like this. So now what happens over here is, so the separation of this chromatid, this part of the chromosome, this part of the chromosome, that takes place due to dissolution of the cohesin protein which attaches these two chromatids to the centromere. When it dissolves, both of them split. When they split, this one becomes separate chromosome and this one becomes separate chromosome. And these chromosomes are said to be as daughter chromosomes and they are said to be as monad. Monad in the sense that chromosome with only one arm. Right? See here, this kind of chromosomes are moving apart each other. This part of the chromosome is moving this way and this part is moving this way. Why? Because these are attached to separate spindle fibers which are continuous spindle fibers. Sometimes they are attached to the spindle fibers along with chromosomal fibers they are attached. So these chromosomal fibers are also there as well as spindle fibers are also there to which the chromosomes are attached. Now what happens? The chromosomes are pulled apart. When the chromosomes are pulled apart with the help of the spindle fibers, especially chromosomal fibers which are arising from the gamete core itself, there are some protein motors are there, and those protein motors, they help in sliding movement of these chromosomes. They help in sliding movement of chromosomes with the help of diamine and use of ATP molecule. Loss of ATP molecule take place and diamine protein is there. It helps in the movement of these or pulling apart these chromatids or chromosomes towards the opposite pole. Is it clear? Now, see here, so this chromosome, it starts to move towards this pole and the centromere is at the forward and the chromatids are following the centromere. Why? Because this is the region where the kinetochore are, so the fibers are there and those fibers are called as what? Chromosomal fibers. So they start to what? So there due to one of the reasons they start to move. One reason is where the proteins are specially present in the kinetochore part, and those proteins I have already mentioned. Motor proteins are there, right? They are dyneins and they transport these chromosomes towards the pole, right? So there are proteins are there, they are called as motor proteins. These motor proteins, dynein is there with the use of the ATP. So protein help in movement of these chromosomes, one thing. And simultaneously disassembly of chromosomal fibers also take place. When the disassembly of chromosomal fibers that take place, that moment what happens, shortening of these microtubules that take place, that is nothing but kinetochore fibers that take place. Whenever they become short, that moment these start to move towards the pole. Imagine every fact over here. Why? Because it is not shown over here. So due to all these reasons, the chromosomal movement that take place. That's why we draw the diagram like this in anaphase. So if it is equatorial line, this is one pole, this is another pole. In case of animal cell, spindle fibers are formed like this. These are continuous fibers to which the chromosomes are hanged over here. See here, this is the centromere, this is the centromere, this is the centromere. It is seen in case of metaphase, but the centromeres they come over here, to which the chromatids are attached. See here, chromosomes are moving in this direction simultaneously. Other chromosomes are moving this direction. So these are moving this direction, these are moving this direction. They are moving totally towards the opposite poles, right? So this is how the rapid movement of the chromosomes that take place in anaphase. And the next stage is the telophase, and it is the last phase of the karyokinases. What happens there in telophase? Let us see now. Next one is telophase. Telophase, it is a long period stage during which what happens, ultimately the chromosomes that reach towards the pole. Okay, so see here, this is the animal cell and the chromosomes that reach the poles over here. The chromosomes that reach the poles over here. This is one chromosome, another chromosome. So totally these spindle fibers are also there itself. Ultimately the chromosomes reach the poles, and these chromosomes they start to uncoil themselves. The condensed thread-like structures, that is chromosomes, they start to what? Uncoil. Chromosomes start to uncoil themselves, forming chromatin network. Okay, these are all chromosomes which are present in the polar sites. Okay, they start to uncoil to form the chromatin network, in the sense that this is the cell in which the chromosomes start to uncoil, forming the chromatic network itself. Chromatic network simultaneously. So the spindle fiber start to disappear and start to come over here at the central axis of the cell. Okay, and simultaneously the reappearance of the nuclear membrane starts. The nuclear membrane that starts to reappear, and simultaneously the nucleolus also start to form. Nucleolus also start to form. How this nucleolus is forming? With the help of what kind of chromosomes, you know? So with the help of the SAT chromosomes, right? So these SAT chromosomes are usually present in 5 pairs in case of human being. Already I have mentioned the names also, 13, 14, 15, 21 as well as 22. So these SAT chromosomes have nuclear organizer region is there. With the help of this one, the nucleolus is found over here, and after that one, after the reappearance of this nuclear membrane as well as the nucleolus, what happens? 2 separate nuclei are respectively formed in a cell. 2 separate nuclei or daughter nuclei are formed in the cell, single cell itself. And this cell undergo what? Next stage, that is cytokinesis. This leads to cytokinesis, means cellular division itself, right? So cell division, how it occurs in case of plant cell as well as animal cell? This is the question. Okay, so this is all about nuclear division. This is all about nuclear division, means 4 stages that we have explained over here. One is prophase, second one is metaphase, anaphase as well as telophase. These are the 4 phases that we come across over here. So this is all about what? The nuclear division. Next one is, that is cytokinesis. Right? So next one is cytokinesis. How this cytokinesis that occurs, so in plant cell as well as animal cell, let us see now. Last stage, that is the telophase. So it is also said to be as a reverse prophase. It is also said to be as reverse prophase. Why? Because it is reversible to prophase stage. What happened in prophase stage? A reverse of that one that takes place over here in this case. So then what happens over here? Let us see diagrammatically. So this is one animal cell. Simply all the chromosomes are so moved to the poles. This is one chromosome, this is another chromosome, this is another chromosome, like this. All the chromosomes are moved to their respective poles, right? They have been moved to the respective poles. These are all spindle fibers that hold the chromosomes respectively, right? So spindle fibers these are, right? So like this, all the chromosomes have gathered in respective poles, and these chromosomes, whenever you make one large chromosome over here, so what happens over here is, this is made up of the chromatin material, that is nothing but DNA as well as so the proteins itself, highly coiled condensed structures these are. It starts to uncoil like this. When it does uncoils, that moment what happens, it becomes elongated, slender, thread-like structure, right? So next, these contains thread-like structures called as chromosomes. They start to uncoil themselves. See here, this is the cell where the chromatic network start to uncoil all the chromosomes over here, and here also it start to uncoil like this, right? So it becomes slender thread-like structure, and this slender thread-like structure around which the reappearance of the nuclear membrane starts. The reappearance of the nuclear membrane starts. So this nuclear membrane is formed from the remains or the fragments of nuclear membrane of the previous cell. So one thing, and even the tubules of endoplasmic reticulum as well as Golgi complex all together form the nuclear membrane around this nucleus. Now, so after the formation of this nuclear membrane, what happens over here is new daughter nuclei are formed over here. Two nuclei are completely formed over here, and simultaneously what happens over here is the nucleolus is also formed over here. So the nucleolus is also formed. So this nucleolus is formed from NOR, nucleolar organizer region. So this is present in the SAT chromosome. Those chromosomes which have satellite SAT chromosome, so they are having a non-nucleoli organizer region. So this region makes what? This nucleolus in respective daughter nuclei. Right? So this is the fact what happens in telophase. Right? So there the disappearance of nucleolus as well as nuclear membrane that took place during the prophase stage, but here it is reappearing. Now two daughter nuclei has been formed. And ultimately this leads to what? This leads to the next stage. What is that? Cytokinesis. It is the cell division. It is nothing but cell division. Now the nuclear division has taken place, and now the cell division is going to be happen over here. Right? So this is about the telophase. It is a reverse process of the what? Reverse process of the prophase itself, right? And one more thing we'll have to remember here. So what is that? The spindle fibers what are here? So here the spindle fibers are there. The spindle fibers in case of plant cell, towards the pole they disappear. Towards the pole they disappear themselves, and the spindle fibers towards the equatorial line, they remain there itself. They remain intact itself. They remain intact. Right? So the spindle fibers they disappear themselves, which are there towards the pole, they disappear themselves. Right? So this is also one more fact that happens over here in this case during telophase only. Simultaneously other cell organelles like Golgi complex, endoplasmic reticulum, all these are formed more and more by a mitosis. It is also kind of cell division what we call it as. So that way many number of endoplasmic reticulum, Golgi complex, such kind of many number of cell organelles are formed towards the pole. So towards the pole all of themselves are formed over here. So like this. So this is the explanation regarding the telophase. And the last one is cytokinesis. Let us discuss now. Last one it is cytokinesis. What do you mean by cyto? It is nothing but cell. Kinesis means division. It is nothing but cell division. It means cell division. So how the cell division that occurs over here in case of animal cell as well as plant cell. So how it differs? Already I have mentioned this now. In animal cell, what happens over here is, if this is the cell, two nuclei are formed over here, inside which the chromatin material is as well as, so the nucleolus, everything is ready now. That recorded. Everything is ready now. Nucleolus is there, and this is the nuclear membrane. So everything is ready. Now what happens over here is, at the equatorial line, the small invagination has been done by the animal cell. Due to the presence of some of the actin as well as myosin, so filaments which are dragging the cell towards the inner side, that is nothing but invagination has been made over here. So and this deepens continuously. See here, if this is the cell, then the deepening of this cleavage or the invagination increases. This is one nucleus, this is another nucleus. When it deepens, that moment all the spindle fibers what are there at the center, all the spindle fibers what are there at the center or on the equatorial line, they gather themselves forming a body called as mid-body. Mid-body itself they form, and this mid-body later disappears, and it leads to the formation of two new daughter cells itself, right? Two new daughter cells are separately made over here. This is one cell, this is another cell. So the disappearance of mid-body that take place, and two individual daughter cells have been made over here. Alright? So this is how in case of animal cell the two cells are formed. But this furrow, what is this called as? Furrow. Or this is also called as invagination. It's not possible in case of plant cell. Then what happens in case of plant cell? In case of plant cell, what happens here is, so the plant cell has a cell wall which is very hard and rigid, and in this case what happens here is, so there is no invagination is possible here. So what happens instead of that one? All the spindle fibers present at the equatorial line, they gather themselves, they elongate themselves and gather all the vesicles of Golgi complex here. So all the vesicles of Golgi complex are gathered at the center, and this is forming phragmoplast. What it forms? Phragmoplast. And this phragmoplast ultimately it reaches the cell wall. It reaches the cell wall. So and these vesicles they release calcium as well as magnesium pectate. What happens here? They release calcium magnesium pectate, and that calcium magnesium pectate itself is responsible for the formation of, this is responsible for the formation of middle lamella. Right? Now the middle lamella is ready. So that what happened here is, so here one cell is formed over here. Here another cell is formed over here. Right? So this forms the cell plate. It is also called as cell plate. the spindle fibers as well as vesicles of Golgi complex all together form the cell plate itself. Cell plate that separates two cells that separate this cell into two daughter cells, right? So this is how in case of plant cell the phragmoplast that leads to formation of a cell plate, and cell plate makes two daughter cells ultimately, right? So there is no possibility of the deepening of the what? So the deepening of this furrow would not take place as if in case of animal cell, right? So this is how the cytoplasmic division or the cell division that take place both in animal cell as well as in case of plant cell. So remaining things we'll discuss in the next class. Thank you.